Okay, welcome to this recording of my BMS bite-sized training Introduction to Statistics session, which was first delivered to the BMS cohort at the University of York on the 30th of May 2023. So as a brief introduction to me, my name is Andrew Mason. I'm a lecturer in cancer informatics and an Elixir data stewardship fellow. The vast majority of my research is all about data, using data in the life sciences, and usually focused on cancer and in avian genomics. So my cancer of choice is bladder cancer in humans. So I'm part of the 100,000 Genomes project. So using multi-omic sequencing data. I also work on oncogenic retroviruses in chickens. And as part of my academic role, I do a lot of teaching and training for undergraduates, postgraduates, and academics through IBRI, the Department of Biology, and as a member of Elixir as well. So my real hope for this session is to give you a better introduction to statistics, get a better idea of the stats to use, some of the common pitfalls, and how you can really make the most of your data. The eventual aim of this is so that you can do better analysis, but also nail stats questions in your Viva. Typically, biologists are not very well trained in statistics and in data analysis. So if you can show yourself to be really, really competent in your understanding of the statistics, you'll do really well in your Viva, and to be honest, you may even know more about statistics than your supervisor. So let's go through some descriptive statistics first. So these are the traditional averages that we all learn at school. So mean, that traditional average of the sum of all your values divided by how many values you had. Median, so the value in the middle of your ordered data set. And mode, the most common. Now, the mode we don't necessarily think about in the biomedical sciences part of research, but it can be quite useful in some other examples like behavioral ecology. I find often that statistics and biomedical work is often quite difficult to visualize. And I often quite like to think about it as I learned it more in a quantitative zoology, quantitative ecology approach. So does a fertilizer have an effect on yield from a field, for example, or are there more animals in this kind of landscape than another? I think sometimes thinking of it that way allows you then to apply it to your cell cultures, to your growth curves, with a better understanding of the underlying assumptions. So these three values, mean, median, and mode, are really important for understanding distribution of data. So the symmetrical distribution, the normal distribution is what we're all really after when we're doing parametric testing. So we'll talk about that later on. But we have other skews in our data as well. So a negative skew, which is a left skew in the data, positive skew, which is a right skew in the data. So in a positive skew, a right skew, for example, there are a higher proportion of large values than you'd expect given the mode value in a data set. So these three values can be helpful in understanding the underlying data set. So mean and median in a bimodal distribution are not very helpful at all. The mode isn't really that helpful either. So the key thing here is always graph your data to get a better understanding of what you're actually looking at and what you can do to understand it. So I'll first talk a little bit about median and distribution using something that we call a box plot or a box and whisker plot. So box plots have a box which is the lower quartile to upper quartile, so that is the middle 50% of your data. You then have whiskers which extend up to the maximum and minimum values, with your median making the 50th percentile of your entire data set. So these box plots are often used to identify outliers in your data, often using this rule of thumb, which is 1.5 times the interquartile range. Obviously, that depends a lot on how variable the data are and whether or not you think outliers in this context are biologically represented. Using these kinds of descriptions, we can do the range with or without outliers, the interquartile range, which describes where 50% of your data are, and 95% confidence intervals. And this 95% interval, it's an error bar that people use, but don't use that commonly. And it's a shame because it can be the first indication of whether you may have groups, if they're normally distributed, which might be different statistically. So going back to the traditional mean, we often talk about the traditional average. This is an assessment really of how accurate, the key thing is an assessment of how accurate we've been in measuring the mean. So when we're looking at these target plots here, each red dot is an individual sample. The plot on the right shows that we've been really accurate in assessing the mean of a population, and because all of our dots are really close together. On the left-hand side, our dots are really spaced out, so there's a lot of variance in the data. So the measurement that we would use to understand this is standard deviation. So how much do the individual measurements differ from the mean value? Low standard deviation gives us better confidence in what we've been measuring. Now, standard deviation versus standard error, this may be the first point in which you start to disagree with your supervisor. So standard deviation is an assessment of how far data points are away from the mean value. Standard error is an assessment of how well your mean values describe the actual population. And so just in the way that I described that then, how well your mean values, this is repeat measurements of different means representing a population. So for example, standard deviation you might use to assess, I don't know, if you're counting the number of nettles in a meadow, you might have 5 different sampling areas. You might count the number of nettles in each sampling area, then you would get a mean value for that meadow. If you then repeated that approach at 4 different meadows, you would have 5 different mean values, each of them with their own standard deviation. Your standard error of the mean would then be an assessment of how accurate each of those means is for describing the distribution of nettles within meadows. Now, the reason that people often report standard error when they should have used standard deviation in papers is it does this. It makes the error bars much, much smaller. But people don't really think about that full name. Standard error of the mean. What is the standard error actually measuring? Standard deviation is individual measurements. Standard error is how well different measurements assess the overall actual mean value. Now, with individual data points like this, even if you report the wrong or the wrong error bars, you can still see the underlying data. The problem is when you have bar charts like this. The standard error one in this context is actually a bit disingenuous because it suggests tightness of the data, particularly if you're trying to use this to suggest statistical significance. It suggests tightness that doesn't really exist. So be really, really careful with this and hold your ground against your supervisor because those small error bars, whilst they look good, are not actually correct. So understanding standard deviation is really key for when we're trying to work out what is a normal distribution. Now, in the literature, you may see normal distributions also being called Gaussian distributions. They're effectively the same thing. So a normal distribution is determined by the mean value. So that mean determines the center or the peak of the bell-shaped curve. And then the width of the curve, so how steep the gradient is on either side, is determined by the standard deviation. Now, there's this quite neat trick here of this ratio 68, 95, 99, and that is 1 standard deviation, 2 or 3 standard deviations away from the mean value. So 95% of the data in this distribution is contained within 2 standard deviations, plus or minus, of the mean. It's actually not quite right, it's 1.96 standard deviations, but the rule of thumb holds out pretty well. This is really important when we're considering this whole significance threshold, the 0.05 significance threshold. This is why this threshold has been used and adopted so widely by the community. So parametric testing needs normally distributed data, well, normally-ish. Normal distribution is a technical requirement, as is equal variance between the groups, so you couldn't fairly compare the difference between the height of daisies and the height of oak trees because the amount of variance in the oak tree group is much larger. The groups should be independent measures, so you couldn't compare proportions of data, for example. And ideally, you're not going to have outliers that massively affect the data. And that's because they would massively affect the normal distribution. If you've got a couple of outliers that give a skew to the data, that can be quite difficult to work with. Now, in practice, these are guidelines. So the first thing that you do when you stick your data into Prism and you test for normality, and it's significantly different from normal, often one of the things you do is panic. Don't necessarily panic. Plot the data, see how different they are from a normal distribution. What can you do to transform the data to improve it? So when we're thinking about parametric testing, it's these terms that you'll all be familiar with. T-tests, ANOVAs, ANCOVAs, Pearson correlations are all parametric tests. We'll come back and talk a bit about those later. So your data aren't normally distributed when you first look. Can you make them normal to avoid having to do non-parametric testing? So data transformations are legitimate. You can do things like a log transformation here to take data that has a right skew and make it more normally distributed. If your data is left skewed, you're going to want to do something where you're reducing the size of that data away. So you're doing things like a square root or a cube root, or you're doing a reciprocal, so 1 over the value. So if you're trying to, in a right skew, you're trying to bring the bulk of the data up. In a left skew, you're trying to bring the bulk of the data down to try and generate these normal distributions. This is completely legitimate. There's a slight error in this plot. So the distribution of B should be with all the data points on the right-hand side, giving this cube root transformation below. So apologies for that error. When you're reporting data, be careful just how you're doing it. So I never like having a log scale. I always find that my brain has to suddenly change it. So a log, it's like if it's a log 10 scale, a value of 2 means 100. I'm having to do that transformation in my head. That's even harder with something like a cube root. That's not something that anyone wants to do on the fly. So when you're reporting the data, maybe even on the axes, put the actual numbers on. So instead of, in that bottom left plot, instead of having 0, 2, 4, you could have 1, 100, 10,000. That makes it easily much more identifiable to the reader what's going on. When you're considering outliers, don't strip them out before you do these transformations, because if there are really, really high values, for example, in data distribution A, they might be brought back into a normal distribution. And it doesn't matter so much when you're doing histograms, but if you're doing bar charts, when you're drawing your error bars, remember that your error bar above will be considerably shorter than your error bar below, because the distances are getting increasingly large for the same actual distance on the plot. So even if after you've done data transformations, it still doesn't look very normal, that's when you can start doing non-parametric tests. Now you want to avoid non-parametric tests if you can, because parametric tests have got much more power, because they're looking at the actual arithmetic difference between points, not just the rank order of different points. So in non-parametric testing, 10 followed by 100,000 they're just 2 ranks apart, compared to in a parametric test, there's obviously a huge amount of actual variance between those 2 data points. But if you need to, non-parametric tests can still be very powerful. Okay, so t-test, we're just going to use this as a logic, and effectively in the t-test, in this case, we've got size of tumor, control, and we applied drug. The idea is has the tumor significantly decreased in size with the addition of the drug? And what we're looking at here is distribution of tumor sizes and whether those distributions are independent. So it's all about this overlap. And it's the proportion of the overlap, which can be found in both distributions, that helps us define a p-value. So by chance, or matching by chance alone, what effect size could we see? Now this all comes in then to our ability to trust p-values. So if you've got small sample sizes versus big sample sizes, that really impacts our ability to do statistical testing. So here I've got a data set I've just created with a mean value of 160 centimeters. It's like a height data set, standard deviation of 10. So if you've only got 50 measurements, you're getting an okay distribution, but there's a lot of noise. Increase that to 500, it's looking much more like a bell-shaped curve. Increase that to 50,000, it fits the bell-shaped curve pretty beautifully. Now the key thing here is that the more samples you have, the tighter your distribution becomes. So eventually, if you have big enough sample sizes, you'll be able to see statistically significant differences. But whether or not that is just a numbers game or biologically relevant is a really key question. So in this dummy example of RNA-seq data, for example, you can see with gene 1, the purple group has a significantly higher expression than green, vice versa for G2. G3, there's no difference. And G4, if you've got enough data points, you might say that these two are statistically different, but are they really biologically different? Same goes if it's down at the bottom. So this is where you've got to be really careful with things like fold change, the magnitude of the effect that you're measuring. Is it really biologically meaningful? So when you're doing your testing, another thing that you'll come across is this idea of a two-tailed test. And that's because of the bell-shaped curve has these 2 tails. Tail going up to the right, all the way to infinity, and a tail going on the left all the way to minus infinity, effectively. So it's two-tailed because it could go in either direction. The idea here in that tumor example from before is that addition of the drug could make the tumor size get bigger, could make it get smaller. Now, there are very few scenarios really where you're able to very reliably say that it's impossible. It could only go in the one direction. So you could think about measuring, I don't know, if you were standing against a wall and then jumping, how far forwards you'd go. It's very unlikely you'd ever be able to jump backwards because you've got your back to a wall. The examples are quite tricky to come up with a good one on the fly, but your one-tailed test is often used in the literature for things like enrichment in something like a chip-seq experiment. And the idea there is that because you're going from nothing to enriched, you're only going in one direction. That's the idea. The problem, though, is that it takes the part of the distribution on the right-hand side, in this case, the 5% shown in part A of the figure, and it makes it bigger, which means there's a higher chance of a false positive. People often use this after the fact. So say if they did a two-tailed test and got a p-value of 0.09 for something getting bigger, then they run it as a one-tailed test and suddenly that p-value is less than 5% because it halves, it goes down to 0.045. Now, that's really, really bad science, but it does get used in papers. So be really, really careful and only use a one-tailed if that is your plan from the start. So that plan from the start all comes down to p-hacking. The idea here is you're fishing for data. You're fishing to try and understand what's actually going on in the data. You're doing loads of comparisons to eventually find something that falls out of significance. Now, the slight difficult thing here is that fishing in the data is effectively what machine learning is doing to try and understand complex data sets. It's doing lots of comparisons, trying to pull meaning from the data. Now, it's one of the criticisms of machine learning that would be a major criticism of your own research for using just doing loads and loads of tests without hypothesis to try and understand the data. Now, if you're actually doing loads of tests because there are lots of comparisons you want to understand, you're going to run into this issue with multiple testing. And because if you use a p-value threshold of 0.05, the chance, if you run lots and lots of tests in a row, the chance of you getting a false positive that's just due to chance increases. So on this graph on the left-hand side, you can see that by 60 independent tests, you're approaching 100% chance that you've actually got a significant result. There are lots of ways that you can try and correct for this. So Bonferroni correction divides your critical p-value, say of 0.05, by how many samples you've got to give you a much more harsh p-value threshold. Things like Benjamin-Hochberg, instead they rank the p-values that you've got to then give you a significant sort of Q-value list, ones that you accept. So we use Benjamin-Hochberg in RNA-seq experiments quite a lot. Bonferroni is quite harsh. These are things to consider in your data analysis. So this is when you get to the crunch of it. So which parametric test should I use? So you can find there are a lot of resources with flowcharts of these things. So if you're doing, if you're looking at the difference between two groups, say with or without a drug, and there's just those two groups, without or with a drug, you can do a t-test. If there's more groups, so say it's without a drug, with drug A or with drug B, that's when you do ANOVA, so more than two groups. And if you're trying to mix continuous and categorical data, so say if you were trying to understand outcomes in an exam and trying to control not just for trying to control for the amount of time people did revision for, that's a continuous variable, and so you do an ANCOVA. Now, people then often come across, well, what is a two-way ANOVA? What's that? A two-way ANOVA is when you've got two categorical variables. So say if it was the example of scores in an exam, your categorical variable, your first question for a one-way ANOVA could be, are boys or girls doing better in an exam? So that's your one categorical variable. Your second could then be something like if they have a pet or not. So that means that you could say, not just does having a pet make you do better in exams, or does being boy or girl make you do better in exams, but you could think, do boys with pets do better in exams. So that could be an interaction term, so understanding the interaction between two categorical variables. Obviously, this can increase and increase and increase, accounting for more and more interactions, a mixture of continuous and categorical data, and that is beyond the scope of this, but you may get examples in your research. So this kind of enormous flowchart kind of covers the parametric data that we've talked about so far. So everything we've mentioned is on this left-hand side, the continuous data on the left. So the section that's all about skewed data, that's when we're getting our non-parametric testing. But first, you would try and adapt your skewed data to go back into continuous. The other side is look at discrete data, so categories, differences between people with different eye color, and then proportions would be differences in things like coin toss tests, like the binomial tests. So the last section I'm going to finish on is correlations. So correlations between X and Y, so this whole thing of correlation is not equal to causation. Here we've got Pearson correlations. So these are the R values that you would see on plots. So R values range between 1 and -1. So 1 is very positively correlated to -1 is very negatively correlated, with 0 being that there's no correlation at all. Now, if you do these plots in Excel, you also get what's called an R-squared value. The R-squared value is as it sounds. It's the R value that you get squared. And that simply explains the amount of variance explained by this correlation. So you can imagine a scenario where you measured your total height and your leg height. They would be very, very well correlated. That's because your leg height is massively explained by your overall height. If your correlation is much weaker, say 0.4, your R-squared value would be 0.16, just showing that only about 15% of the variance in your data set is explained by that correlation, because there's a lot of other data that's also there. There are also other variables which are clearly important. Now, the key thing here is that these all show the same gradient. Now, gradient isn't necessarily related directly to correlation, because the gradient is all about magnitude between your different variables. So again, this is where you have to link into what's biologically significant versus what's just a very, very good correlation. The cautionary tale with this is this group called the Anscombe's Quartet. So Anscombe was a mathematician who wanted to better understand data and how it's described, how it's visualized. So in this, all 4 of these plots, the data sets have got the same mean, same standard deviation, same Pearson's correlation value. But you can clearly see that these data do not, well, most of them aren't even linear. So you shouldn't do Pearson's correlations with these data sets. With the 3, so the top left is the one where you could do a decent Pearson's correlation. The rest of them, you're probably going to want to consider whether there are outliers or whether there are sort of multinomial features that you'd want to look at, or you'd want to go down the non-parametric route, such as a Spearman rank correlation. So where should I do my statistics? So places like Excel and Prism, they're fantastic, as are online tools such as the Social Science Statistics website. If you're going to be doing these things regularly, I would really recommend investing time in R or Python, particularly if your data sets are going to get bigger. Obviously, it takes time to learn. Like any language, you need to be doing it all the time. So just think really, how often will you be doing stats like this? So as a little summary, just some QR codes here to existing resources. This presentation, including this recording, will be available at that QR code. A previous course I've done on an introduction to C Bioportal with Elixir is at that QR code if cancer sequencing data is something you're very interested in. And sequencing data in general, I've covered in this series of data management bites on sequencing data. Okay, so hopefully this was a good introduction to statistics and describing your data. Key things really are to graph your data, take a look at the data, describe it before you start picking your stat testing. It's all about whether or not you can get the most out of your data, and if you can do that in a reliable, robust way. Thanks for listening.